I'll start by defining what mobile genetic elements are, and basically they can be defined as any part of the genetic material that can move around the genome or between genomes or between bacterial cells. So it's a rather loose definition, but there are a lot of very different types of mobile genetic elements, and I'd like to try and include them all in this definition so that we get an appreciation of what they are and the breadth and depth of things that they can do. Just to give you an overview of what I'm going to talk about in this talk, I'm going to discuss the various types of mobile genetic elements. I'm going to talk a little bit about their role in the spread of antibiotic resistance and their role in modulating and spreading virulence genes. And then I'm going to end the talk by just talking a little bit about how they've been used extensively in molecular biology. And I'm going to talk a little bit about some new work that we're doing to try and use them in stopping the spread of antibiotic resistance. So to begin then, so the different types of mobile genetic elements, to start on this slide from the simplest, working our way down to the most complex. Insertion sequences are probably the simplest of all the mobile genetic elements, and they just encode enough genetic information to move from one part of the genome to another. They're completely selfish elements, and they don't code any other function other than their own movement. The next level up in complexity is the transposons, and these are genetic elements that can move in the genome, but what differentiates them from the insertion sequences is that they often contain accessory genes that are not involved in their own transposition. So quite commonly they can encode antibiotic resistance genes, but they can encode lots of other things as well. And more complex still, we come to the plasmids. These are a very heterogeneous group of genetic elements, but their main defining feature is that they have an origin of replication which allows them to replicate distinct from the bacterial chromosome. And they again can spread antibiotic resistance genes and virulence genes and can conjugate between different bacteria. Some can and some can't. And then we've got the conjugated transposons, which are also called integrative conjugative elements. And these are probably the most abundant of the conjugative elements in bacteria. And these normally can't replicate independently of the bacterial chromosome, but remain integrated into the chromosome, but can excise and sometimes replicate as well. So there's a bit of crossover really in the properties between plasmids and conjugated transposons. And conjugated transposons now are also called integrated conjugated elements, or ICE. And then finally, I've included bacteriophages in this definition of mobile genetic elements because they can move their genomes between different bacteria. They're viruses of bacteria, and they can predate on bacteria and destroy them and replicate themselves, but they can also integrate into the bacterial genome, and on occasions they can replicate like plasmids. So I think I want to include them in the types of mobile genetic elements because they can also move genes around as well. So I think it's fair enough to include them in this definition. Okay, let's show this in cartoon form. This is a slide that I took from Adam Roberts, our ex-colleague, and I like to include this in talks because it nicely encapsulates what mobile genetic elements do and their heterogeneity. So here we've got a bacterial cell in cartoon form, and this is in green is a bacterial chromosome, nothing on it at the moment. And we move up again from complexity like the previous slide. The simplest of the mobile genetic elements are the insertion sequences. These tend to be quite small, less than 1 kilobase. And next are transposons. These are a little bit bigger because they've got some cargo genes, like antibiotic resistance genes. And next is conjugated transposons. These can be quite big. They can be anything from 18 kilobases right the way up to hundreds of kilobases in size. And there's mobile introns as well, which are another type of genetic element that can transpose and move around the genome. But they don't usually have much of an effect on genomes because they're the ultimate parasites or commensal really, because when transposons or conjugated transposons insert into a gene in the genome, they can knock that gene out. But introns don't do that because they're on transcription. The intron RNA is spliced out to get a fully functional messenger RNA. So they're kind of the ultimate mobile element in some ways, because they have no effect and they can just hide in genomes and move about without causing much of an effect. So they're probably not as well studied as transposons and conjugated transposons. And I'm not going to talk much more about them in this talk. And then we got the extra chromosomal elements like the bacteriophages that I was describing. Sometimes bacteriophages, instead of destroying the cell, as shown here, can integrate their DNA into the bacterial chromosome and replicate happily along with the chromosome. And we got the plasmids shown here. These replicate independently of the bacterial chromosome. And all these genetic elements can interact with each other. Shown here, the IS elements can transpose to different parts of the genome, and they can transpose into plasmids. Transposons can do the same thing, and it's kind of interaction. How's that? Get back now. Carry on. Conjugated transposons cells can transpose into plasmids, and again the introns can do this, and sometimes plasmids can integrate into the bacterial chromosome. And phages also can sometimes behave like plasmids. They can integrate, they can inject their DNA into the cell, and sometimes again, instead of destroying the cell, they can replicate like plasmids. And phages can also transduce DNA, so they can package bacterial DNA and transport it to other genomes on occasions. So all these genetic elements are part of a continuum. They can all interact with each other, and they can all do things that you sometimes think a particular class of genetic element wouldn't do. So I just wanted to show that slide to show how these things all interact, and how there's not always a hard and fast definition of any particular element. And as I was intimating there, some mobile genetic elements can themselves promote horizontal gene transfer. So what is horizontal gene transfer? Also called lateral gene transfer. So vertical transmission is when a bacterial cell, shown here, divides and passes its genetic information on to its two daughter cells, and this is called vertical transmission, and this happens when cells divide and pass on. But horizontal gene transfer is when you get transmission in one direction from a donor bacterial cell to a recipient, and this is called lateral horizontal gene transfer. And this is probably very important for bacteria. It's probably at least as important as vertical transmission. And there's various mechanisms of horizontal gene transfer in bacteria. As shown here, this is a recipient bacterial cell. There's transformation in which naked DNA is taken up by a recipient cell. There's conjugation, which is probably the most important mechanism, that is shown here, where a plasmid or a conjugated transposon encodes the genetic information required for its movement from a donor cell to a recipient. And there's also, as I was intimating before, there is transduction, where a phage packages bacterial DNA instead of its own DNA and can move that into the recipient cell. Okay, so let's move on to talk about conjugated transposons in a little bit more detail, because this is what I spend quite a bit of my time working on, and as I said in the abstract, I was going to illustrate these things from my own research. So conjugated transposons then are widely distributed. They were first discovered back in the 80s by Don Clue and his co-workers, and they can transfer between different types of bacteria. There are many different types now, and they often encode genes that allows host bacteria to exploit particular ecological niches. This is quite commonly things like antibiotic resistance. And more recently, these elements have been called integrated conjugative elements, or ICE. So you often hear them referred to as ICE, or sometimes conjugated transposons, but they are more or less the same thing. I'm just going to show a little bit about how these elements actually move. This is taken from a review by Abigail Salyers. So this is a conjugated transposon inserted into the bacterial chromosome. This is a bacterial chromosome here. This is the conjugated transposon. What happens is these elements excise and form this circular molecule. This is the transposition and conjugative intermediate. And when they transfer, you get cutting as a region on the transposon called the origin of transfer, or ORIT. So this single-stranded nick is made, and single strand is transferred from the donor cell into the recipient through a mating pore. So this requires intimate contact between the donor cell here and the recipient. So the next step is a kind of replication where you get second strand synthesis in the donor and second strand synthesis in the recipient. So now you've got a double-stranded circular molecule in both donor and recipient. And the next thing that happens is that we get insertion of the conjugated transposon back into the genome of the donor and into the genome of the recipient. So the element is transferred from donor to recipient. And this is a typical conjugated transposon called TN916, which was the first one that was discovered by Don Kluhl, as I said, back in the 80s. And these elements are typically modular in that they've got a region required for conjugation. And these genes, shown in red, are the genes required for conjugation. And these include genes that encode regions for cutting the DNA at that single strand, then push that single strand across from donor to recipient. They encode genes for holding the two cells together, and genes for making that, genes encoding proteins that make that mating pair. They also contain a region that allows them to integrate and excise from the recipient chromosome. So here we've got an excisionase, which is required for excision, and an integrase gene, which is required for integration. So they're proper little genetic engineers that can move themselves about and get in and out of genomes using these various genes that they have. And they also often contain a region, an accessory region. In this case, in TN916, it's the gene encoding tetracycline resistance, but it can be anything really. So this is the cargo DNA that's transported around when these things move, and they contain a regulatory region because obviously it doesn't pay for these elements to be continually excising and transferring if they don't need to, if there isn't a suitable recipient around. So these regulatory regions encode genes that can basically assess the state of the cell and whether or not the cell is in proximity of recipients, and which will make it desirable for the element to excise and transfer. Now, TN916, like genetic elements, are one of the most commonest elements found in bacteria. And this is taken from a review that I did with Adam a few years ago. And this just shows the different types of genetic elements and their general modular feature. They all have these regions involved for conjugation. They all tend to have the regions involved in integration and excision. But they contain a lot of different cargo regions. This is TN5397 that we've worked on for a while. But the other elements from found in different types of organisms contain different antibiotic resistance genes. These ones from Klebsiella contain resistance to mercury and other antibiotics. So they're quite heterogeneous, and they can acquire antibiotic resistance genes and other genes, and they can move them around between very distantly related bacteria. So they're quite important in helping bacteria evolve and adapt to different situations. Now, I want to move on to talk about mobile genetic elements in Clostridium difficile, now called Clostridialis difficile, which we've worked on here for a long time. And this is an organism that's a pathogen of the gut, and it causes diarrhea in hospitalized patients, often antibiotic-associated diarrhea. So if patients are given antibiotics, it disrupts their gut flora. It allows C. difficile to take over and cause disease, and sometimes it can cause quite serious disease called pseudomembranous colitis. And this is a picture of a gut from a patient with pseudomembranous colitis, and you see these horrible yellow pseudomembranes, and when it gets to this stage, it is pretty life-threatening. And the two major virulence factors of the organism are two large toxins called toxin A and B, and the genes for these elements are actually been shown to be capable of transferring to non-toxigenic strains. That's why I'm including this story in a talk about mobile genetic elements. And a lot of C. difficile mobile elements are conjugated transposons, responsible possibly for promoting the transfer of the toxins, but certainly for promoting transfer of antibiotic resistance determinants. Now, as I was alluding to, the toxin genes of the Clostridium C. difficile are capable of transfer, and the toxin genes are on a genetic element called the parlock, and the parlock encodes the two major virulence factors of the organism, and it's important to note that non-toxigenic strains do not normally contain the parlock. So this is the C. difficile pathogenicity locus, or parlock, on the top line. It contains these two large genes which encodes toxin A and toxin B, and in addition to that, it encodes this gene TCDR, which is an alternative sigma factor which is involved in positive regulation of this region and involved in crosstalk with the rest of the metabolic state of the host. The role of this gene is a bit more controversial, but it's thought it's a negative regulator of toxin production, and TCDE is thought to be a bacterial holding which allows export of the toxin genes from the cell. And the reason I put these other two things on from C. sordelii, which is a related Clostridia, and C. perfringens, is to show that toxin regions like the C. difficile parlock are found in other Clostridia, which also gives some clues that they might actually be mobile. So this was work done by Mike Brouwer a few years ago when he discovered that it was done here at the Eastman. This was, he discovered parlock transfer, and this was discovered by accident really, because we were looking at transfer of this conjugated transposon called CTN1. So he was looking to see if we could transfer CTN1 from this C. difficile strain 630 into this non-toxigenic C. difficile strain called CD37, which has been used in the lab for a long time. And CTN1 had an erythromycin resistance gene on it, so we could select for it. And selection was made on erythromycin and rifampicin because CD37 is resistant to rifampicin, and this strain is sensitive to rifampicin. So the only strains that could grow were erythromycin, rifampicin resistant strains. And so we found that transconjugates had this CTN1 as we were selecting for it, but some also contain the parlock, and that hadn't been selected for at all. So the parlock had transferred without selection from this toxigenic strain to this non-toxigenic strain when you were just selecting for CTN1. So this was quite a surprising finding really at the time. So what it did indicate was that parlock and conjugated transposon transfer are linked. So we found that the parlock and conjugated transposons both transferred at a frequency of just one transconjugate per 10 to the 8 cells, which is a very low frequency of transfer. But if the transfer of CTN1 and parlock were independent, we would expect a transfer frequency of 1 in 10 to the 16, 10 to the 8 times 10 to the 8, which you just would never see. So it's obvious that, and what we did see was that 1 out of 9 transconjugates had the parlock when selecting for the CTN, and 2 out of 7 also had the conjugated transposon when selecting for the parlock. So obviously there's some form of linkage between transfer of the parlock and the conjugated transposon in these experiments. So how to explain this then? Well, we did this with the help of genome sequencing. So this is the whole genome sequence of C. difficile 630, which is the donor, and you can see that the parlock is located here, CTN1 is located here as shown, and there's another conjugated transposon TN5397. So there's a number of conjugated transposons that are dotted around this chromosome, and some of them are near to the parlock. So the next thing that we did then was sequence the transconjugates, the CD37 transconjugates, and these are shown here. So this is superimposing the transconjugate genome on the donor 630 genome. So let's look at this section where the transfer of the polyp is blown up a bit. So here we've got 7 independent transconjugates, and this DNA here represents 630 DNA that's transferred to CD37. So CD37, remember, doesn't have these conjugated transposons and it doesn't have the polyp. So we're looking at 1 independent transconjugate here, and you can see a large, over 300 kilobases of DNA is transferred from 630 to CD37, and this includes the polyp. These all include the polyp because in this experiment we were selecting for the polyp. But you can see that different size fragments transferred in each of the different transconjugates that were picked independently, and some of them had TN5397, but I think this is because TN5397 is transferred on this large fragment. Some of these also had another conjugated transposon called CTN5, which transferred at the same time and again wasn't selected for, and some of them had this and some of them didn't. So what we think is happening is that all these conjugated transposons and these large genomic fragments are using the same pore to transfer from donor to recipient, but they're not all transferring at the same time. I hope that makes sense. If it doesn't, we can obviously talk about it a bit more in the question and answer session at the end. So let's try and explain what we think is happening with polyp transfer then. So if you remembered in the earlier slide I showed you a conjugated transposon excising and circularizing, and then we get the cut at the RET and a single strand is transferred from donor to recipient. So what we think is happening in the case of polyp transfer is that the conjugated transposon, a conjugated transposon, we're not quite sure which one yet, is not excising, but we're still getting the cut at the RET shown here. So this is the donor cell. We get cutting at RET, this is the recipient. Here's a cut at RET, transfer of the chromosome this time to the recipient. So the whole chromosome is actually transferring because the conjugated transposon isn't excising. So if it doesn't excise and you get this cut at the RET, instead of transfer of just the conjugated transposon, you would get transfer of the whole chromosome, and this fits with what we've actually seen. So now you get a single strand transferred. Because the donor and the recipient are quite related and the DNA sequence apart from the polyp is probably identical, we can get homologous recombination. So the region where the polyp, or the region where there wasn't a polyp, is the region flanking it. I'll show you this in the next slide. It's probably easier than me waffling on over this, but what happens is that as this region gets transferred, we get recombination, and a bit of the chromosome is now replaced by the incoming DNA. And let's show that on the next slide. It's how that refers to the polyp. So if the top line is the recipient genome, you get these two genes, CDU1 and CDD2, flank where the polyp normally is. So here's a strain, toxigenic strain. Here you see these two genes on either end of the polyp. So this is the recipient chromosome. That single strand that I was telling you about containing the polyp comes in. Here's all the polyp, and these regions are identical, so we get homologous recombination, and effectively what happens is this region containing the polyp replaces the this small non-coding region, and essentially now integrates into the genome of the recipient strain. So this region now is outside, and as it's not in the genome, it has free ends, and these are degraded by exonucleases. So essentially, we've converted a non-toxigenic strain to a toxigenic strain. And now, what's the biological significance of this? Well, one thing is you obviously get you can get non-toxigenic strains being converted to toxigenic strains. But at the moment, people are using and have used non-toxigenic strains as a treatment or as a preventative from infections with Clostridium difficile. So this really needs to be looked at a bit further, because if you're transferring non-toxigenic strains into a host that has toxigenic strains, that strain that you're using to cure C. difficile could be converted into a toxin producer. So this probably needs to be looked at in a little bit more detail. So just to summarise this little section of my talk, I hope I've shown you that any gene is potentially capable of transfer. However, the frequency of what this occurs differs depending if the gene is on a conjugative element. Obviously, if it's on a discrete conjugative element, then it will transfer at a high frequency. But we can also get transferred regions like the pollock, which are not on any discrete conjugative element, but they've just been mobilized by other integrative elements in the vicinity. And obviously, well, transfer isn't driven by the selective pressure, but the success of the transconjugates is driven by the selective pressure of the environment. So I'm sure this has a big effect on natural selection. Certainly all this shuttling of genes gives natural selection a lot of raw material on which to act. So let's just summarize what we know about the role of conjugated transposons on the evolution of bacterial genomes. Well, they can often carry genes for accessory functions, probably the most important of which being antibiotic resistance. When they insert into genomes, they can inactivate genes. They can sometimes target specific domains. I've not talked about this in this talk, but there are some conjugated transposons are site-specific and they can insert into certain targets. And also, as I've just been showing you, they can mobilize large genomic fragments. I'm just going to talk very briefly about the role in phage in virulence in C. difficile and in other organisms. Phage have got a big role in virulence. And on occasions they can transduce antibiotic resistance genes, and also in the case of C. difficile, a number of other workers have shown that phage can modulate and can both up and down regulate transcription in the pollock. So phage as well is important as these other mobile genetic elements I've been talking about. So then, I hope I've shown you that mobile genetic elements are central to bacterial evolution, and they've got a huge impact on human health and well-being because they spread antibiotic resistance, which, as we all know, is a modern scourge, and they can also spread virulence factors and other genes that allow bacteria typically to adapt to new environments. So to move on to the next part of the talk then, the mobile genetic elements are not all bad, because they are the absolute basis for modern molecular biology. Plasmids, for instance, are absolutely central for gene cloning, and all these transfers, integration, excision, encoded by mobile genetic elements, provide an absolute wealth for genetic engineers. With millions of years of evolution that can be exploited to manipulate almost any organism, not just the prokaryotes that you want. And also, the defensive system that bacteria have evolved to prevent them being exploited by mobile genetic elements can also be used. For instance, restriction enzymes are a defense mechanism used by bacteria, and they are now central in molecular biology methods, as I'm sure you all know. And more recently, clustered regularly interspersed palindromic repeats have been adapted for gene editing, and these have developed an absolute revolution in molecular biology. And I'm going to talk a little bit about these now. This is taken from a review by Jiang et al. And this just shows how CRISPRs work in quite a simple way. So they were initially discovered by these regular arrays in the genome by genome sequencing projects. And what was found was that you've got repeat region. These repeats are shown in grey here. and these regions were interspersed by spacers. But the interesting thing that was found in these spacers is that they were homologous to phage genes and plasmid genes and other conjugated and genes that found in other conjugative elements. So this gave a clue to what they were, and it is in fact turns out that they're an immune system against these genetic elements. So what happens in these arrays is that if you get an abortive infection by a phage, as shown here, say this phage doesn't actually kill the cell, but the phage DNA is broken up, these Cas genes can catalyze, encode proteins that can catalyze the integration of these spacer and phage genome fragment into the CRISPR array. So as we see here, the CRISPR array increases by one spacer, by one repeat and one spacer, because it's integrated this DNA from this phage. So it remembers attack from this phage. So if it sees this phage again, it's kind of immune to it, as we'll show in the next part of this diagram. There are different types of CRISPRs, but I'm only going to talk about type 2 because this is the one that's been used mostly in genetic engineering and synthetic biology. So this is so-called the adaptation phase, in which new invaders or fragments of new invaders are incorporated into this array. And next is the CRISPR complex biogenesis, which is shown here. So you get transcription of this sequence, and you get this long RNA molecule, and you get this transacting RNA molecule, which is a cofactor in the enzymatic reaction to destroy invading DNA. And these form these double-strand RNA regions with the spacers. Sorry, with the repeat regions, not the spacers. And eventually this is matured and cut down. So we eventually get this molecule, which is a protein RNA complex. So this is a protein, Cas9 protein in this case. Here's the spacer region, and this is the repeat region with its transacting RNA. And this now can hunt down invading DNA. So if it sees a sequence in this invading DNA, which is shown here, which is homologous to this spacer RNA, and if there's the protospacer adjacent motif, which has to be there, so this is usually a sequence of 3 base pairs, which is just next to where the homology lies, and this is absolutely required, because if it didn't recognize this protospacer adjacent motif, then this riboprotein would cut the DNA of the CRISPR array in the host, so it'd actually kill itself. I mean, it's a way of recognizing invaders and recognizing self. So again, we get this sort of thing in the immune system. So anyway, it sees this invading sequence, it will bind and it will make double-stranded cuts, and that would result in this invading DNA being destroyed. So how is this used then? Well, you can use this in genetic engineering, and how is this used is shown here. So this riboprotein makes these double-stranded breaks, and that will kill the cell generally, or will certainly destroy the genome where these double-stranded breaks are made. But you can add editing regions to this. So if this region is targeted, you can get a process called non-homologous end joining, which is not common in prokaryotes, but it's often found in higher organisms, and this is used to manipulate eukaryotic organisms. Or you can add a template of your own choosing, and if this donor template is homologous to these regions, we can get repair by homologous recombination. So if you chose this sequence here, you remove the target of the CRISPR-Cas. So if you remove the target of the CRISPR-Cas will kill and destroy every genome except the one that's got this target that you want. And so this allows only the edited genome to survive. So it's a way really of precisely removing all the genomes that don't have the bit that you edited, because editing, you can edit one genome in a million, but your CRISPR-Cas will kill all the ones except the ones you've edited, and then you can end up with your edited genome. This is an incredibly powerful tool for molecular biology, because you can choose. It's only 20 base pairs is the targeting region of that RNA, and I'll show you this in the next slide why this is so powerful. This is a typical targeting construct for bacteria. So you can make a little synthetic CRISPR array to do your targeting, and all that's required for this is a gene encoding the Cas9 protein, the transacting RNA, and the repeats here, and the spacer. And you can choose whatever spacer you want to put here, so you can virtually target any gene for destruction, because there are many different sites in almost all genes that you can use, that you can target using this array, using this, it's called a guide RNA, but it's basically a mini CRISPR array with a spacer, sorry, with repeats here in white, and the spacer, and you can choose what spacer you want to target whatever gene you want. And so what they've done in this little experiment is that you also need an editing template, because this region will guide this Cas9 transacting RNA to destroy this genome. But if you add an editing template, you see which part you want to destroy, and you see which part you want to change. So you can change this sequence now so that it doesn't contain this target, and this region won't be destroyed. So if you transform this into a bacterial cell, for instance here they've added kanamycin resistance. So if you transform this into a bacterial cell, and you transform the editing template, and this is the gene you want to change, and you transform those all into a cell, the only organisms that will survive are those that have got this edited gene. So it's an incredibly powerful system, and it's actually relatively easy to change the targets, because you just need to change this 20 base pair sequence. So I'll just finish off now by saying a little bit of the work that we're trying to develop at the moment is to develop this system to specifically target antibiotic resistance genes on mobile genetic elements. Now, as you probably all know, antibiotic resistance is one of the biggest global, well, it used to be until COVID, one of the biggest global problems, and there's a risk of post-antibiotic era coming. So we designed this, well, before I go into this Limbot system, as we call it, this synthetic gene cassette, let's just see what happens when you have a population of bacteria, shown here, and you treat a patient, and these are antibiotic resistance plasmids shown in red, and you treat a patient or an environment with an antibiotic, you're just left with antibiotic resistant organisms. So the use and abuse of antibiotics has led to an explosion of antibiotic resistant organisms. So all these organisms now are resistant after you've treated them. And people have tried using phage and various things to target antibiotic resistance genes and get rid of antibiotic resistance. But the problems with phage is that they're very specific to certain types of bacteria, which in some circumstances is good because you don't damage the normal microbiota. But we're trying to develop a system which we called a Limbot. So I won't go into the details of it because we're currently trying to get this funded. I don't want to tell everyone how it works, but in simple terms, we just divide this system. We're using CRISPRs to actually target antibiotic resistance genes, so they will remove all these antibiotic resistance genes from an environment or move them from mobile genetic elements carrying antibiotic resistance genes. So hopefully this will be a promising technique for ridding the world of antibiotic resistance. So, I mean, this system is not just limited to removing antibiotic resistance and virulence genes. It can remove virtually any genes you want from microbiomes, and there's probably a great deal that you can do. there probably is an end to the potential of this approach, but it's a very big, it's an end that's perhaps a long way off to be used for microbiome engineering. So to conclude then, I hope I've shown you that mobile genetic elements are responsible for the spread of antibiotic resistance and virulence genes, and mobile genetic elements and their components are the cornerstone really of modern molecular biology. Without these elements, it really wouldn't be possible to do modern molecular biology. And I'd just like to acknowledge a few of these people on this list for stimulating conversation and for doing a lot of the work that I've been talking about in this lecture. So at this point, I'll try and unshare the screen, and I'm happy to answer any questions.